And then we'll finish that up probably Monday, and then we'll go back a little bit to whole plants. So the last lecture, I always cover the plant part and then cover the chemistry part because that seems to make a little more sense. And we'll be talking about water potential, these different terms that we use, and what we're building now is the actual physics of how water moves into and out of the plant. And what in the world is that? Okay, now, this is a really famous picture. You see these all the times. Where was this picture made? I think it says on there. It doesn't. Where's the only place you can make a picture like this? This is water. This is on the shuttle. And you remember from general chemistry that hydrogen bonding is really weak, right? Well, no, it's not. Compared to when you have two carbons sharing electrons, it's weak. But the movement of water from the soil all the way to the leaf is totally dependent upon hydrogen bonding. So if you take water and you squirt it out where there's no gravity, then the water binds to water. It will, by hydrogen bonding. And he could stick his hand in this water and actually move it around. And it would move around as sort of a warped sphere, and then it would form a sphere when you stop moving it. If you put water in a Teflon pail and there was no gravity, you could stick your hand in there because your hand, of course, has hydrogen bonding capability, and you could pull out the entire bucket of water. So we get this idea from chemistry that, yeah, it's a weak bond, but it's really, hydrogen bonding probably has more to do with the movement of water than the other thing. Now, the shape of the cell, and you've heard this time and time again. At any given time, if the plant has water, water is going into the plant, it's moving up the plant. If it's during the day, there's a lot of transpiration, and in every single cell, water is being continuously pumped in. And when the water's pumped in, then the balloon, the vacuole, and then the balloon inside the balloon, the water's going to fill up. The cell membrane is going to go press against any open space. So if it's a square box, the cell membrane is going to press against all the possible sides. And you get to the point of equilibrium where for every water molecule that is pumped in, one water molecule is pumped out. And that's, you guys know that's turgor pressure. And if you look at a leaf, this carpet is coming up. If you look at a leaf in the middle of the summer and the leaf is wilted, the turgor pressure is 0. There's not enough water going into that leaf. Give you an idea how important that is. Last year, no, not last year. Year before last was when we had the very severe drought. And in most of our grapes are grown around Lubbock. And Lubbock is, so we had really hot weather, we had very little water, and Lubbock has this constant wind. If you've ever been to Lubbock, you know. So with the high temperatures and the wind, it was not possible to put enough water into a plant for it not to wilt. So we had a lot of problems with grape yield because even if with best irrigation you could have, because of the high temperatures and the wind, the transpiration was coming so fast that the leaves were just basically always partially wilted. Usually that doesn't happen. Now, let's manipulate water. Anybody ever make eggplant parmesan or anything? Why do you take your eggplant, you cut it in slices, and you usually either put it in salt water or you actually put salt on it? What are you doing?
Trying to desiccate it.
You're desiccating it. You're basically moving water from inside of the eggplant to the outside of the eggplant. You guys know about osmosis, and we're going to talk about water potential. This is something you should write down. No matter what, water always moves from a higher concentration of water to a lower concentration of water. Always. So if you take an eggplant and put it in salt water, there's more water inside the eggplant than outside the eggplant, so the eggplant loses water. Okay? And if lettuce wilts, and this is routinely done in restaurants, they take the lettuce and they dump it in water, because if the lettuce wilts, it means the water concentration is lower, and so you put it in water so that water will go in. In that case, water is moving from a higher concentration of water to a lower concentration of water. Okay? And so this is something that, is routinely done. Actually, I think I showed this slide before, but I want to go over it again. Is what I want to hammer in is what water does. In this case here, you have really nice cells. These are cell walls, primary cell walls. And in this case, this leaf has been put in a solution of sucrose. Let's say it's 10% sucrose. So 10% sucrose has a lot less water than the cell does. So what's going to happen if you put a leaf in 10% sucrose? Which way is the water going to move? It's going to move out. And so now you see the plasma membrane shrinking, and it forms this complete protoplast, remember, the living cell, that's completely been removed from the cell wall. And you may have done this in high school, or you may have done this in freshman biology. And you can take this leaf and you can put it in water, and the cells will fill up and they'll be turgid again. Then you can put it back in a sucrose solution. The cells will shrink. You can do this over and over again, until basically it runs out of energy because it's not carrying out photosynthesis or anything. But you can do it several times. Okay? And plant cells have to be able to do this. So only when you get to this point, we'll talk about later, where it's so wilted, the protoplast is so small that damage occurs and it can't recover. Okay? And in the right, this is the right side, right? Okay. Here is a protoplast, and what has been done on the right here is that the cell wall has been enzymatically removed from the cell. And you put the protoplast in a sucrose solution, and so you have a living cell there without a cell wall, a living plant cell. And in this case, if you know anything about GFP, these are green, two different fluorescent proteins where they're looking at the location of, I can't remember what it was, but just this is done a lot. And you can take that cell and you can actually get that cell to regrow a cell wall. Now, 99% of the times, you throw the cells away. What you're doing is trying to find a particular protein, which is not part of what we're talking about now, just to show you that nice picture. A lot of times there are probes that cannot get through the cell wall. The cell wall is a real barrier to anything but water. So if you want to find a protein and localize a protein using antibodies or whatever, or a GFP or whatever, you've got to take the cell wall off to get that protein in sometimes. Now, let's go over things that you've probably had before about why water is water. The thermal properties are such that water is pretty much going to stay water. If I put a bucket of water here and I heat it up, then I am going to have some of the water that's going to vaporize. But it's still water. It's still H2O. Doesn't break down. So if I set a container of water here, I come back next year, it's still going to be water unless it's evaporated. It's not going to break down to protons and hydroxyls. There's always a little bit of that, but it's never going to just spontaneously break down to protons and positive protons and negative hydroxyls. Water stays water. And we've seen some condensation reactions, and there's also hydrolysis reactions where water is actually added. Water is clear. So again, back to cell structure. If you're looking at a tree, what is the only place that is green? And the answer is not chloroplast. Where is the only part of that tree that's green? Be a good question for the exam, actually.
It's going to be at those little pigment reaction centers.
The grana, or the thylakoids. You could say the thylakoids too, but you're not even looking at the chloroplast. The only part of that thing that's green are those green stacks, this grana. That's where most of the green is. And then, of course, water is used to move water around. That's what turgor pressure is. Because of the shape of water, let me go ahead and go to the... You all remember this from general chemistry, is because of the molecule, the electrons are not equally shared, so a given electron at any given time is going to be usually found around oxygen more than the proton, right? So this is a polar molecule with the oxygen being negative, and then the protons being relatively positive. And then at that cartoon at the bottom, it's showing you then that the oxygen is going to bind, or it's going to be attracted, the negative to the positive, negative to positive, positive to negative, and that's what hydrogen bonding is. So if you turn over, and I've actually got a compound, if I can remember to bring it down, that you can't pipette. It has no hydrogen bonding at all. So you can pull it up in a pipette, but as soon as you stop suction, it just falls right back out. You can put your finger over the pipette, and it has no bonding, no hydrogen bonding to the glass at all. It just falls right back out. It's sort of weird looking. It's hard. The only way you can actually measure it, you can pour it into a cylinder and measure 10 mils, or you can weigh it out. You can't pull like a mil up in a pipette. It doesn't work. It just comes right back out. On the other hand, what in the world keeps happening? Oh, I was still taking the exam, I guess. Okay. So, a couple other things. High specific heat, high thermal conductivity, heat of fusion. When do we take advantage of the heat of fusion to keep plants alive? Anybody know? If we spray water on plants that are just right at the freezing temperature, when the ice freezes, it actually produces a little bit of heat. So if it's just going to freeze, so in the spring here, and I have some pictures of Dr. Burns, well, not Dr. Burns, Tim, actually putting up these temporary sprinklers with PVC pipe because it was already blooming. They were starting to bloom, and there was going to be a freeze. Now, not a hard freeze, but a freeze. And so if you spray water, and the water then protects the plants, first, the fact that the water's there gives it a little insulation. But if there's actually a little freezing, then it actually liberates a little heat. So it's actually a positive thing. Now,
They did that with citrus orchards.
yes, they do that in citrus orchards, yeah. And if you're just going to freeze, this is not going to protect you against a hard freeze. And they also use helicopters just to move around, to move the air around. Used to use smudge pots, which have been outlawed a long time. Used to it'd be just these pots that would be every 20 feet or so that produced really black smoke, and which would heat it up a little bit. So physical chemical properties, I'm just going to just throw these up just to show you the differences. We all know what the properties of water are. The density of water is highest at 4 degrees C, which you guys know, and there is some heat required. It's called heat of vaporization, which is how much energy you have to put in to take a liquid molecule and convert it into a gas, okay? So you have the solid ice, you have a liquid, and then you have a gas. But it's always water. That's the thing. It's not, water doesn't break down to protons and hydroxyls. Okay? Now, how are things solubilized? And this is important. If I take salt, which is a crystal, and I put it in water, immediately when it comes into contact with water, then the sodium and the chlorine atoms dissociate completely, totally. And they do that because they're completely surrounded by water. So what you're looking at is something that is solubilized. And so in plants, we have all sorts of salts that are soluble, okay? If I put a sucrose molecule in water, or if I put sucrose in water and I shake it up or whatever, then what I'm doing is I'm separating those sucrose atoms or sucrose molecules and putting water between them, okay? We can go to the other extreme, and if I take oil and water and I shake it up, it's not soluble, right? It may look like it is, but basically water and oil do not mix. And you guys know you can shake up water and oil, set it down, come back, and the water and oil is going to separate. So something that's soluble is completely surrounded by water molecules. And if you're talking about tea, then you know if you use actual sucrose, you know you got to stir it like crazy because it's very close. You know, it's a very cold temperature, so there's not much energy there to separate the sucrose molecules apart. Right? So if you use sucrose in tea, you tend to put a lot more sugar in it than you actually use, because if you look at the bottom of it, it's still, you have a layer of sugar that big on the bottom, or that thick on the bottom. Okay? This is very important because now, if we've done this, we now have a solution of water that is less than 100% water. And of course, everything inside us, inside plants, is less than 100% water. We're mostly water, but we're not 100% water. And what we're going to talk about in plants is the fact that the differences in the water concentration of water, and I know it's a backwards way to think about it, but the differences in the concentration of water is why water moves. Okay? Now let's talk about some terms. Cohesion is a term that we use when we're talking about hydrogen bonding of water to water. The same. Cohesion, water to water. Okay. So if I take a pipette and I pull it up, pull water up in it, a big thing is that water comes up as a continuous stream. It doesn't come up one molecule at a time. It comes up because of cohesion. You know, and when you turn a hose on, water doesn't move through the hose one molecule at a time. It moves as a continuous stream of water molecules. You know, we talked about this last lecture, I think, about how water moves in plants. The tensile strength is something that I mentioned last time. And if you could grab water, you know, and you see this, I think I was saying, you know, you see these documentaries about how a plane crashed or something and a piece of metal fell. They'll take the metal and they'll pull it out until it finally breaks apart. That's a tensile strength, of course. And water has a tensile strength. So if you could grab water at the top and bottom end and start pulling it out, you would have to pull a bit of a force before it's separated. You're never going to see that, of course. You can't do that with water. And then the other term, surface tension, let's not worry about that much. And the other term is adhesion, and that is the fact that is why water will move up in a pipette. The water molecules hydrogen bond to the glass. Cohesion is water to water. Adhesion is hydrogen bonding to something else. So I was telling you about this liquid, which I can't remember the name of it is. So I pull it up in a pipette, and as long as I'm pulling the suction, I'm pulling the molecules up because they're attracted to each other, and then they just fall right back down. They have no attraction to the glass. So water, you pull it up, you cap the pipette, water stays up because water is attracted to the glass, or if it's a xylem cell with all the secondary thickenings, the water hydrogen bonds to the lignin, the cell wall, because of adhesion. So you got two really important things. You've got water moving with water because it moves as a stream, and then it also sticks to the side. And you see this, you know, when you get a Coke or you get something that has a straw in it, you notice how there's always a little bit of liquid, a little tiny bit of liquid above the surface? That's because of adhesion. The water is adhering to that. Like I said last time, you know, they prick your finger, and you got that really, really thin capillary. The blood pulls up. The blood is being pulled up because of adhesion and cohesion. Okay? Okay, and this is a figure from your book, and the whole point of this is to show you, like I've been saying, that at any given time, if I put a bucket of water in here, some of the water is going to go into a gas form, but it's going to go in and out as water. It doesn't break down. It's incredibly stable. Now, how water moves? Bulk flow. This is the way water moves through a garden hose. This is the way water, one of the ways that water moves in the xylem. It is by bulk flow. It means the bulk of water. Is that water does not move as one molecule at a time, but moves as a continuous, you know, almost like it's easier to understand if it were ice moving. Of course, ice wouldn't move, but it's easier for you to say if I have a xylem and I can pull the ice up. So that's basically how water moves, as, you know, a continuous column, a continuous solid mass of water. The other one that we're going to talk about, of course, is diffusion, and that's from higher to lower concentration. Here's the classic one. We have the movement of water. We have a solution on the left, and again, the concentration of water on the left is lower than on the right. And in this case, we've got a hole between these two compartments. And so what's going to happen is water is going to move this way, and then these particles are going to go that way until everything is at equilibrium. Okay? And this is not what happens in cells. What you'll see, what we'll be talking about, the fact that at any given time, there's always a difference in concentration of water between any part of the plant. And part of this is due to the absolute concentration of water. Part of it's due to a vacuum effect, you know, pulling water up in the xylem. And And then if we talk about then water potential, remember we mentioned that last time, then the water potential is highest in the soil and it's lowest in the leaf. And it's more negative in the leaf. Remember, the water potential of pure water is 0. So the soil might be 0.2, or the cell could be 0.3 or 0.4, minus 0.3, minus 0.4, and the leaf is going to be minus 500 or something. Okay? And that gradient is from the soil all the way to the tip of the leaf, if the plant's doing okay. Now, you all know what osmosis is. It's the movement of water. Through what? A permeable membrane. The classic one is water can only move through the membrane. You've heard of reverse osmosis, probably. So with osmosis, osmosis only works if we have a membrane, and cells have membranes. So we have membranes that allow for the movement of water, but not solutes. Now we have the same diagram here, except this time we have a membrane in place. Okay? What did I tell you about the movement of water? Which way is water going to move? So where's the highest concentration of water? The right side, right? So the whatever is solubilized in the water can't move. So water is going to move from pure water, a higher concentration of water, to a lower concentration of water. So it's going to move, right to left. I don't know why I can't get that today. And what you see then is something that's unique, is that we now have this side actually moving up. So we're creating a force there. Does that make sense? So if we go back here to this one, they're going to be the same. Now just think about this. I think on the last slide in this, there's actually, so water is moving here. It can only move here. And guess what? The water's always going to be at a higher concentration here than on this side. So what happens then is we increase here, and this will move up until the force of gravity is equivalent to the force that's being placed on the movement of the water. So this can't go up forever, right? So you could, if you wanted to do it really slowly, you could make a car lift, where you have pure water versus some kind of a lower concentration of water, and you're creating actual force there. You're pushing things up. That's also how root pressure works. Remember last time we talked about root pressure? Does that make sense? So we always have then water moving here until the force of gravity limits how far up it can move. And now something that's really, really important is that means then every time one water molecule moves in this direction, this force is going to force a water molecule to move back. Okay? So that's in equilibrium in terms of force. And in plants, that's what equilibrium is. It's the pressure of the cell membrane pushing back when it's completely filled with water. So you put one water molecule in, you pop one water molecule out. Okay? And the height there is limited by gravity. And that's why we cannot explain the movement of water up by root pressure alone. Because it moves up to a point, and then since this is a continuous piece, these molecules can't come apart and move on up, then the gravity then determines how high it can go. Okay? That's very important. Now, Let's talk about the cell. And we have a lot of membranes. We've got a plasma membrane, and then if it's a mature cell, we've got a vacuole that's almost all of the cell, and then we have organelles. So you've got all these membranes interacting, moving things across them, and what we're going to talk about mostly in here is just movement of water in terms of from the plasma membrane into the cell, okay? Cell membrane, and then the tonoplast, the vacuole. In a mature cell, the vacuole is 99% of the space. And let me just finish up here with, goodness, go back. Which is what you're seeing here. Here's the vacuole. So in this case, a water molecule is moving in and forcing everything against the cell wall. Okay, and we will finish with there. Any questions? Okay, I'll send an email out in a few minutes.